Hello everyone, welcome to our third tutorial video on AI-powered denoising. In the first video we talked about supervised learning. In the second video we talked about unsupervised learning. In this video I will explain how to apply these methodologies in the real world. I will describe how we trained deep neural networks to remove noise from electron microscopy data in order to reveal the dynamics in the atomic structure of nanoparticles that are important for catalysis. Let's get to it. I'm going to show you how to learn how to denoise using deep neural networks, which can lead to scientific discovery, and we're going to focus on electron microscopy. Our scientific motivation is to study catalysis. 90% of all manufactured goods involve catalytic processes somewhere in their production chain. Catalysis is extremely important for energy, pharmaceuticals, materials, transport, and environmental applications. In many of these applications, nanoparticle-based catalysts are crucial. In order to understand the functionality of catalysts that are based on nanoparticles, we need to observe the changes in the atomic structure of nanoparticles. But this is actually very challenging. It's very challenging because atoms are very tiny. Here I'm going to show you a real picture of a platinum nanoparticle, and what we're interested in is how these individual atoms are rearranged as the nanoparticle interacts with other materials. But these atoms, their order of magnitude is 10 to the minus 10 meters, which is 1 Armstrong. Remember that 1 nanometer is 10 to the minus 9, and hence the name of nanoparticles. Electron microscopy enables us to visualize materials at this microscopic scale by shooting electrons into the material and then gathering the resulting data in sensors from which we can afterwards see the atomic structure of the material that we have imaged, that we have shot the electrons at. The problem is that if we want to use this technology to observe the changes in the atomic structure of the catalytic nanoparticles, we have to gather data very, very fast because the atomic structure changes very quickly on very short time scales. So we want to gather the data as fast as possible. When we do that, unfortunately, I forgot to say, new, relatively recent electron microscopy sensors enables us to acquire data at the timescale of milliseconds, which is very exciting because this could potentially enable us to observe new dynamics. The problem is that when we acquire data so quickly, the signal is very weak because there are very few electrons hitting the sensors so that we can barely see what is going on. So here I promise you, and you will see later that this is true, that there is a supporting material here, which is cerium oxide, and on top of it, there's a platinum nanoparticle that is sitting there, and then there's vacuum. But just by looking at it, you could hardly tell. As I said, part of the problem is that there's very few electrons hitting the sensors, so you could think of increasing the number of electrons, so shooting more electrons at the material, which, if we're getting fancy, is called increasing the electron dose. But the problem is that if you do that, you will fry the material, you will completely destroy it, and here the whole point is seeing how the nanoparticle structure evolves, and if you fry it, then you're not going to be able to see that anymore. So there is a dire need to denoise this data in a way that enables us to see what is happening underneath all of this noise. If we apply traditional methods to this type of data, they don't work very well. So as an example, again, here I have a picture where there's this catalytic nanoparticle. Okay, that's what we're zooming on here. And we're interested in seeing its atomic structure, but we basically see very little from the raw data. If we apply traditional linear shift invariant denoising, which is essentially Wiener filtering, you might have heard, or Gaussian deblurring, we are able to identify the periodic patterns, but we actually don't see where the nanoparticle ends and where the vacuum begins. So here, there's actually vacuum. There are no atoms, but this linear denoising method hallucinates the atoms because it has picked on the periodic patterns. It's not able to see the boundary. Non-linear denoising methods that were available before deep learning do better. So here you see that we no longer hallucinate as much in the vacuum, but we're not really able to see the precise atomic structure of the nanoparticle, which is what we're interested in. In contrast, deep learning works extremely well. Let me show you the data that I had shown you before. And as promised, indeed, we see that there is a platinum nanoparticle here sitting on this cerium oxide support, and now we can see the individual atoms very well. Thanks to this technology, my collaborator Peter Crozier and his group at Arizona State University, they were able to identify previously unobserved dynamics in platinum nanoparticles, and we reported these findings in this Science article. So I will refer you to the Science article for more details, but I do want to show you a few examples. So, for example, they observed displacements of atomic planes within the nanoparticle, which are called shearing. So here, some planes of atoms within the nanoparticle are changing. Also, they observed add layers forming on the surface of the nanoparticles. This is when there's an atomic surface that is attached to the nanoparticle, which can be important for its functionality. And apart from that, they also observed rotations of the nanoparticle. So I want to emphasize that some of these changes in the atomic structure occur much faster than the time scale of milliseconds, but milliseconds is already faster than was previously possible. So at least you're able to glean some of the atomic reconfigurations that are happening within the nanoparticle, which is very exciting. Okay, so between these two frames, the nanoparticle rotated. And finally, my favorite one, which is this fluxional behavior, where the atomic structure of the nanoparticle becomes completely destabilized. We see how essentially it melts within the image, which is very cool. Again, for the scientific details about all of this, I will refer you to the Science article. But now what I'm going to talk about is how we managed to apply deep denoising to enable the visualization of those dynamics. As often happens in the real world, we had to apply different strategies until we could make this work, and they actually used the two main paradigms for deep denoising that we explained in the previous videos. So both supervised learning and unsupervised learning. As I will explain afterwards, we also combine the two paradigms in a semi-supervised framework, and we also had to care about evaluation, which can be very challenging in the real world. Okay, so that's the plan for the rest of the video. We already did a whole video on supervised learning, so I will refer you to that for more details. But in general, the framework of supervised denoising is that we learn the parameters of a convolutional neural network that maps noisy data to a denoised prediction by comparing that denoised prediction to clean data and minimizing the loss, which is typically the mean squared error between the denoised prediction and the clean data, or rather the clean images. And we do that over a large training set until the neural network is able to denoise the images in this training set. The problem for our real-world application is that this requires access to clean and noisy pairs. So given that out of the electron microscope we only get this super noisy data, what do we do? Well, what one can do is one can simulate the types of images that we expect would be coming out of the electron microscopy data. So this shows a platinum nanoparticle, again on a cerium oxide support, because we do know the type of materials that we have put into the microscope. We can use simulation software to compute the 2D projection that would be observed in the electron microscopy under certain imaging parameters and depending on the orientation of the nanoparticle. And this enables us to obtain simulated images that would be clean images if there was no noise in the electron microscope. We actually know the type of noise that is to be expected, which is dominated by shot noise. So it's just IID Poisson noise at each of the pixels. So we can take all of our simulated data that was simulated in this way and add noise to this, noise to the data to obtain these clean noisy pairs that then enable us to train a neural network. We presented this framework in this paper below. And now let's take a look at how it works. Our first lesson that we learned while trying to make this work is that if you use a traditional denoising architecture such as DNCNN, which is just a convolutional neural network that we describe in more detail in the video on supervised learning, it doesn't work well. The reason is that it doesn't have enough field of view. It's not using enough pixels, or rather I should do it here. It's not using enough pixels to denoise to produce the denoised, each pixel in the denoised output. Instead, we need to use a unit that has like a multi-resolution architecture, which enables it to have a much larger field of view. We talked about this in more detail in the supervised learning video, so I will refer you to that, but this is actually quite important. It allows us to produce good denoising, at least on the simulated data. So these are results on the simulated data. The method worked extremely well. But when we move on to the real data, the problems, as always in the real world, the problems arise, right? So here is data again from a platinum nanoparticle lying on a cerium oxide substrate. This is a reference that we obtain for this specific real data because we know that the nanoparticle is not changing very much. So here we can just do temporal averaging to get a reasonable reference and at least see how the method is working. Of course, what we're really interested in is in seeing how the atomic structure changes. But this is just a sanity check. And in our sanity check, what we saw is that the atoms on the actual nanoparticle were recovered very well. But if we look at the support is not well recovered. So I'm not an expert in electron microscopy or material science, but what was explained to me by our collaborators is that this pattern here is actually not present in the denoised image, which is problematic. The reason for this, we discovered, was that in the training set, images without this pattern dominated because the imaging parameter space that we were considering for our training data was too large and did not adequately represent the imaging parameters that actually happened when the real data were actually acquired. In order to address that, we redesigned the training set essentially by selecting a subset that was more relevant to the real data, and that actually managed, doing that, we managed to recover the proper structure in the support. But this reveals a very important shortcoming of the supervised denoising framework for real-world applications, which is that you kind of have to know what you are expecting in order to produce a relevant training set, which can be very, very constraining. Because of that, we moved on to trying out unsupervised learning, where you can train neural networks using only noisy data. We have a whole video about different ways of achieving this, so I won't go into a lot of detail. I'll refer you to that video. The method that worked better for our electron microscopy data was blind spot denoising, where the idea, I will explain the idea very quickly. We get a neural network that maps noisy data to a denoise prediction, and then we compare that denoise prediction to the noisy data. So you will say, this sounds very strange because the neural network doesn't need to denoise to do very well here, right? It can just output the noisy data. But the catch is that we don't allow the network to see each. So when we're trying to denoise a specific pixel, we don't allow the network to see that noisy pixel in its input. Okay, there's this blind spot architecture where you are only using the remaining pixels, the remaining noisy pixels, to denoise that individual pixel. And the only way you can do that is by approximating the signal if the noise is independent between pixels, which is approximately true for our electron microscopy data. That we're going to see how it worked in the real data. And the exciting thing here is that then you can just use real data to directly train the network. So you don't have to worry about the types of structure that you're expecting. You can just let the neural network do its thing and reveal the structure to you. So again, we have the same data and the same reference as in our as in the results for our supervised denoising framework. And what we observed is that if we just let the unsupervised learning proceed, it will eventually overfit the noisy data and produce artifacts when we try it on new held out data. In order to avoid this, we realized that we have to monitor the denoising performance of the neural network on held out data. And specifically what we monitor is this blind spot cost function that we use for training. So we monitor that on held out data and use it to stop before the overfitting occurs. And if we do that, the method actually works very well. In particular, remember this issue with the supporting structure, the structure of the supporting material. We actually recover that very well. The only caveat is that depending on how much noisy data is available, sometimes we see the atoms on the surface of the nanoparticles a bit less clearly with unsupervised denoising. But again, here we don't need to simulate anything. We're just training the networks directly on the real data. And this framework was introduced in this paper, and it relies on prior work on unsupervised denoising that we mentioned in the unsupervised learning video. Because of this issue where we realized that the unsupervised denoising method was actually working very well, but had this issue where sometimes the surface of the nanoparticle was not super clear, we were inspired to try out a semi-supervised learning framework where we try to combine the best of both worlds. So in supervised learning, the paradigm, as I explained before, is that you have a lot of clean images and you train the neural network. These are the parameters of the neural network that are learned, basically by adding noise to these clean images and making the neural network denoise them. Then at the end, you take your real data and you apply the neural network. In the unsupervised learning paradigm, you actually use real noisy data to train the network, and then you just apply the network to denoise. But the problem there is that you might not have a lot of real noisy data, or within the noisy data that you use to train the network, there might not be enough diversity in the structures that are represented. You might want to make sure with your with simulated data that you are actually exposing the network to certain features that might appear when you actually apply it. So what we thought of was combining these two paradigms, using a lot of simulated data to pre-train the neural network, if you want, using the simulated cost function, and then after that, fine-tuning the network with real data. When we applied this on the real electron microscopy data, at first, and this is again exactly the same data as before, where we have this plasma platinum nanoparticle on the cerium oxide, and we have this reference because this specific nanoparticle is not moving around, so we know it's we can just find its atomic structure by averaging across time. What we observed is that if you try to fine-tune all the parameters, it's disastrous. It completely overfits the noisy data. But then we realized that if you only fine-tune a subset of the parameters of the network, this actually works very well. And the subset that we fine-tune are the gain parameters within the batch normalization layers of the network. And I will refer you to the video on supervised learning for what batch normalization is. But essentially, these gain parameters are used to scale and shift the features within the convolutional layers of the neural networks. It's a small fraction of the total parameters of the network, and that's what prevents it from overfitting when we do this unsupervised fine-tuning. More details in this paper, Adaptive Denoising via Gain Tuning. The semi-supervised learning framework is called Gain Tuning. So now we have seen how to apply supervised, unsupervised, and semi-supervised learning to our real electron microscopy data, but we still have a problem, which is how to evaluate it. Because typically in the denoising literature, life is great, because what people do is they look at their denoised output and they compare to clean images, because they have these synthetic data sets where they're adding noise to the images themselves. But of course, we cannot do this with our electron microscopy data. We only have noisy data. So how do we know how well we are doing? Well, we came up with this new technique for evaluating in an unsupervised way, which uses a cost function, which is essentially the main idea underlying most unsupervised denoising methods, where you take your denoised output and you compare it to noisy data of some kind. And again, I'll refer you to the unsupervised denoising video for more details. And this is not enough. This part of the cost function that we use for training. So this is the cost function that we use for training. That is not enough to evaluate, because it's not an accurate estimator of the supervised mean squared error. And the reason is because there's an extra term that appears. This analysis is in the supervised denoising video, so I will refer you to that. But you can actually correct that term using additional noisy frames. And when you do that, you obtain an unbiased, consistent estimator of the mean squared error between the denoised image and the clean image, assuming that the noise is independent across frames. And this is very important. So not here, sorry. Independent among the noisy frames, and also assuming that you have the same atomic structure in each of the frames, which is not always exactly true in practice, but we hope that it's close enough. This method was introduced in this paper. Using this evaluation, we determined that a blind spot unsupervised denoiser that used several frames was better than one that used only one frame. So here you see the difference for a platinum dataset and a cerium oxide dataset. We also determined that it was better than this neighbor-to-neighbor method, which we mentioned in the video on supervised denoising, and much better than linear shift invariant denoising using a Gaussian filter, which, I mean, as we saw before, the visual difference between these methods is quite remarkable. So thanks to the unsupervised evaluation, we can know how we are doing on average on noisy datasets and determine what method is better for which dataset. However, it's important to emphasize that the mean squared error, so this is at the end of the day the sum of squared errors between the denoised image and the underlying clean signal, may not capture scientifically meaningful structure. In particular, the absence or presence of atoms might change the mean squared error very little, but it's completely crucial for our scientific application. Because of this, we developed a method where we estimate the likelihood of the observed data given that we have detected certain atoms, essentially to quantify the consistency between the data and the hypothesis that the atoms are there. This was introduced in this paper, and I'm going to just show you a quick example of how this would work. You have your data, you denoise it, and then you decide where you see individual atoms. And now we know that the noise is approximately Poisson IID, so we can compute the likelihood of this data, given, for example, that an atom is here and has a signal intensity that can be obtained by just averaging the pixels that we assign to that atom. If we do that, we can obtain a likelihood per atom that we can then visualize. And what we see is that in parts of the denoised image where it's unclear whether there's an atom or not, you can see whether the presence of that atom is consistent with the data or not. In this case, that atom that seems to appear is not as consistent with the data as the rest, and this is simulated data, and it turns out that that is a hallucination of the neural network. So in this case, this likelihood map enables us to identify this hallucination. That said, evaluation remains a very, very important challenge in real-world applications that is definitely not completely resolved. So what's the conclusion here? The conclusion is that AI-powered denoising can reveal the hidden structure in electron microscopy data. Supervised, unsupervised, and semi-supervised methods work, but you actually have to be careful about how you apply the methods, as we saw from the lessons that we learned along the way. And overfitting is always looming, and something that you have to be very careful about. And finally, evaluation is really a major challenge. For more information, I refer you to our tutorial, Learn, Denoise, and Discover, a guide to deep denoising with an application to electron microscopy. And I want to highlight that a lot of the methods that I mentioned here were developed by Shreyas Mohan, so a shout out to him. And I also want to highlight that Peter Crozier is really the scientific mind behind this whole project. Here you can see him beside his electron microscopy at his lab in Arizona State University. So please take a look at his work for more information about the electron microscopy and material science aspects of this. And I would like to end by acknowledging support from the National Science Foundation, without whom all of this work would have been completely impossible. Thank you very much.